other words, when you've got that, here's the very first, here's that order, the very first. You look at that very first picture of the kidney. That's when your brain identifies that, and you say, oh, that's kidney. And it might not be this low, it might be the next one, but your brain says, oh, that's kidney. And then everything you've learned about that by looking at the higher magnification slides and listening to the DVD races through your brain. So you say, for example, oh, that's kidney. And okay, I know there are kidney tubules in there, there are renal corpuscles in there, the corpuscles are divided into Bowman's capsule and a glomerulus. They're both made of simple squamous. The glomerulus function is diffusion. Bowman's capsule is filtration. The tubules function is all that's racing through your brain. And so then when I get to a higher magnification picture, like maybe this one, well, you already know it's kidney. And now you know immediately those are the tubules, and you know they're made of simple cuboidal, and the function is selective reabsorption. You wouldn't want to start with a high magnification. You wouldn't have any perspective. It's kind of like if we're traveling. If we're going to Daytona, we're not going to start, and we're leaving from Colorado and we're driving, we're not going to start with a map of Daytona, are we? We're going to start with a map of the United States, because we've got to decide how we're going to get to Florida. We may go up to Lyman and get on I-70 and head out across Kansas, Missouri, and then go south, or we may go south and cut across Texas and Louisiana. How are we going to get there? Low magnification, what do? You get a large field of view. As you increase the magnification, you get more detail, but you get a smaller and smaller field of view. So again, your initial identification very often occurs with the lowest magnification. When I test you, since I know that, when I test you, I usually start with a low magnification. So you've identified this as kidney. We don't want to start with this. I mean, most of you could tell that was kidney, but it's not a place to start. It's interesting, if you think about it, how often in the world does somebody pick up a piece of tissue off the floor and say, what is that? Oh, that's the spleen from a Bulgarian monkey, 3 years old, on the island of Huskov. Okay, in forensics you do that perhaps, okay? But generally you know this is a slide from the lung of a male, age 26, no history of smoking. Is the tissue normal, precancerous, or cancerous? You normally know what you're looking at before you look at it. Well, you guys don't. You know, it's more like, pick a number. So, think about how the microscope works. We start with low magnification. That's where we get our bearings. That's where we typically identify the organ. You don't want to start with this. I mean, you can probably figure out what it is, but by starting with the low magnification, you know it's a cross-section of the small intestine. And everything you know about the small intestine, everything you've built into those books is racing through your mind. And so as I increase the magnification and formulate the question, you're ready for it. The other thing I want you to keep in mind is that most of what we know and most of what we see, we know and we see because people have studied the tissue with much more sophisticated microscopes. Again, think about our microscopes. Think about your basic, lab 1 in biology. This is a microscope, the arm, the base. This is how you carry it. This is the rotating nose piece. What's the total magnification of a light microscope? 1,000 times life. And that's with the oil immersion lens, which nobody on earth uses except in microbiology classes or advanced biology classes. Because if you don't clean the oil off the objective, you ruin it. So other than Biology 204 at Pikes Peak Community College, the highest magnification you ever have is 500 times light. 500 times light. What are you going to see with that? Not much. But people with the electron microscope are able to view things up to 1,000,000 times light. And they take pictures and they write books and we read those books and we see those pictures. And so we learn what's in there. They can see Golgi, they can see ribosome. We can't see any of that stuff. What do we see? We see the nucleus. That's about what we see with our limited little cheesy light microscope. Why don't we have an electron microscope? Well, it would fill this room and cost several $1,000,000. And it's so sensitive that if it was put, actually it wouldn't fit in this room. We'd have to increase the area of the room and make the ceiling about 10 or 15 feet higher. But if we put one in this room would have to be on a foundation that wasn't connected to the rest of the building. Imagine that you're looking through a microscope right now and what you're looking at is magnified 500 times light. What happens if somebody puts their hands down on the table? The vibration is magnified 500 times. Well, imagine you're looking at something magnified 486,000 times, and you got people walking above you. It doesn't work. So, why don't we have one? Well, they cost millions and millions of dollars. They take ginormous rooms. The foundation has to be disconnected from the rest of the building. There's an awesome video in the library if you want to go see it, and what you can do is, 3 or 4 of you that study together can go in there and watch it in one of those rooms, but it's called epithelial tissue. There's another one called connective tissue, but if you just watch one, I think it's a mere 30 minutes. But a guy named David up at University of Colorado in Boulder goes through all the epithelial tissue, and in 30 minutes he gives you vast amounts of information. But the coolest part about the video is it starts out with him showing you an electron microscope. It's worth just watching the video to enlighten yourself about what an electron microscope truly is. As we go through histology, we'll learn a lot about several systems in the body. Histology actually serves as the introduction to several systems in the body. It doesn't do us much good to say, these are renal tubules, they're made of simple cuboidal, the function is selective reabsorption. You've got that in your books already. This is a corpuscle, outer Bowman's capsule, inner. You've done all that. What's it mean? Selective reabsorption. What does that mean? Okay, that's what we want to learn, and so we will. And for us, it'll be an introduction to renal physiology or an introduction to the urinary system. When we look at the small intestines, it doesn't do us much good to say, well, the small intestines are convoluted into villi. The villi are lined with simple columnar. There are 3 types of cells there. And in the middle is the well, so what? What's it mean? What's it do? How does it work? So we get an introduction to the urinary system. We get an introduction to the digestive system. And we take this stuff with us forever. Not just into 202, but into patho, into medical surgical nursing. And you'll add to these books next semester, specifically when we go over the histology of the endocrine system. So it's a huge, huge, very, very important foundation. Several times I'll have to clear the board and draw a picture, not necessarily an anatomical picture, but a diagram to explain things, because again, our goal here is to learn how the stuff really works. So, as you know, this is kidney, and you know the structures in there. What we want to do is understand how they really work. If we zoom in on some of these things, they are renal corpuscles. And actually two renal corpuscles are shown in this picture. You've got the inner glomerulus, the outer Bowman's capsule. The other structures in the picture are tubules, renal tubules, which are shown here. You can see very nicely that they're made of simple cuboidal. The other thing that will happen as we go through some of this, and it was happening with some of you who I won't point out because it's certainly not my goal to embarrass you, but it was happening in the lab on Monday. I had some people that actually were amazed to realize that that's one cell. I don't know if they thought this was a cell or what, and it doesn't matter, but the person was asking me, why is it cuboidal? I don't see the cubes. And I drew a little diagram, and I said, well, here's one of those tubules diagrammatically. And see, those are the individual cells. That's why it's simple cuboidal. So these are the individual cube cells. So there's some light bulb will come on as we go through some of this. If we compare the cuboidal cells there to these cells, these are squamous cells because they're very flat. They're wide and they're flat. Here's a really prominent one. Here's its nucleus. But so what's going on with all of this filtration, diffusion, selective reabsorption? So if we talk a little bit, actually I may need you to come. Thank you. You're welcome. Functional units of the kidney. Nephrons, just like the alveoli, are the functional units of a lung. Nephrons are divided into renal corpuscles, which are divided into Bowman's capsule, glomerulus. Those are simple squamous. The other part of the nephron, renal tubule, which is simple cuboidal. The renal tubule is divided into 5 specific parts. I'll spare you their names until next semester, so we'll pick up right here in that context. So what's this all about? Your kidneys filter your blood. It's like talking about your lungs are for gas exchange, but the lungs are divided into millions of alveoli. They're actually where the gas exchange occurs. If we look at a kidney diagrammatically, and we focus on this specific function here of the tubule, of selective reabsorption, there's a kidney containing millions of nephrons. I'm using red for a reason, so make sure you do as well. Blood goes into the kidneys. Blood comes out of the kidneys. These nephrons millions of these things in there. These nephrons have the ability to keep what you want to keep and get rid of what you want to get rid of. Like a filter. You have a filter in your furnace, your air conditioner. Well, it doesn't trap the air, it lets the air pass through because you want to keep it. But it traps the dirt, the dust, the bugs. If you have a spa or a hot tub or a swimming pool, it has a filter. The filter doesn't trap the water, but it traps the gunk. So selective filtration is what this selective reabsorption is all about. So for example, in your blood, you have glucose, you have amino acids. Well, those are valuable. You want to keep them. So they're in the blood that goes into the kidneys. But they're kept in the body. So they're also in the blood that comes out of the kidneys. Now, something that you want to get rid of leaves the kidneys through another tube called the ureter. You've heard of that. You know the ureters are made of transitional epithelial tissue. So something in the blood that you don't want would be urea. Urea is waste. In fact, birds concentrate their urea into a white paste that you've seen on your car. We don't concentrate our urea. We eliminate it by diluting it with water. So these functional units of the kidneys, the nephrons, millions and millions of nephrons in each kidney, have the ability to detect what's in the blood that we want to keep. So it's in the blood that goes into the kidneys. We want to keep it, so it's in the blood that comes out of the kidneys, versus what should we get rid of. So we send it out through the ureter. There's, of course, a urinary bladder here. And eventually a toilet. That's what the T stands for. Selective reabsorption. Let's break that down. Okay, everybody knows what absorption is. I've spilled some water. I'm going to use paper towels and absorb the water. Reabsorption. I'm going to absorb it, but I'm going to put it back where it was. Where was it? It was in the blood. It moved into the nephrons. If I want to keep it, I need to put it back into the blood. So to reabsorb is to put it back into the blood. So using my glucose example, which is in the blood, the glucose is now in the nephron. Because I want to keep it, I reabsorb it, meaning put it back where it was, which is to put it back in the blood. What if I don't reabsorb it? Where will it end up? In the ureter. So if I don't reabsorb it, if I put my waste here, I don't want to reabsorb it. If not reabsorbed, it is lost as urine. So I don't want to reabsorb my waste. I don't want it in the blood coming out of the nephron or out of the kidney. So my waste is lost. Reabsorb, put it back where it was. Selective reabsorption. Do I want to reabsorb everything? The waste that's in the blood and moves into the nephron. Do I want to reabsorb the waste? No. I want to lose the waste. So I selectively reabsorb. I reabsorb things I want to keep. I don't reabsorb things I want to lose. To reabsorb, requires the kidney tubules be permeable to the material. In other words, how can I get it out of the nephron if the nephron is impermeable to it? It's stuck in there. It's like a dog in a fence. But can a mouse get out of the fence? Yeah. So the nephron is permeable to some things, impermeable to other things. And then next semester, when we really build on this, we'll start to understand how hormones can affect the permeability of the nephron to water or to salt. All of this in front of you, I've got to clear the board. So just a diagrammatic view of the urinary system. Nothing really new here to you. You have two kidneys. That's where your nephrons are. The ureters, transitional epithelial tissue, conduct the urine from the kidneys to the urinary bladder. Also transitional epithelial tissue. Then the urinary bladder drained by the urethra. This particular urethra is not really, well, it's certainly not male. It's not even quite female. It's just sort of a diagram. If we look at a kidney, so it's in here where all of those nephrons are. If we blow this up so that we can actually see a nephron, so everything that's not red is the nephron. So there's material in the blood, say glucose. It goes in. We want to keep the glucose so it comes out. How did it get out? Well, it moves from the renal corpuscle into the renal tubule. As the material travels through this renal tubule, if it stays in there, it goes to the toilet. So if you want to keep it, some part of this tubule, and again there are 5 parts there, some part of that tubule has to be permeable. So if that part of the tubule is permeable to glucose, the glucose moves out of the tubule, is reabsorbed, meaning put back where it was, put back in the blood, so that the glucose that was in the blood here is in the blood coming out. The waste stays in the tubule and is given up to the toilet. So 2 words, selective reabsorption. but 15 or 20 minutes to understand what those two words mean, okay? Your next slide or set of slides are the ones that come from the lung, functional units of the lung alveoli. This is sort of an intermediate magnification of lung tissue. As you know, the function is gas exchange, simple squamous, thinnest tissue in the body, specialized for gas exchange. If we blow that up, there we have a couple of alveoli. But what's meant by gas exchange? Again, it doesn't do you any good to memorize the function. Memorize the name. Name this organ, lung. Name these structures, alveoli. Name this tissue, simple squamous. Give the function, gas exchange. That's absolutely worthless. Absolutely worthless. But coming into the room this morning with that knowledge allows you to build on it. We can't talk about the lungs and the alveoli without introducing the cardiorespiratory systems. Okay, I've just linked two systems. You know from introductory material, this is a systemic class versus a gross class. All introductory classes use that approach. It's the way to make it manageable. But as we proceed, continuously think about linking systems. If we're studying movement, it's and I know I look ridiculous. It's musculoskeletal, because my muscles make my bones move. Musculoskeletal. But what makes my muscles contract? My nervous system. So it's neuromuscular. So it's neuromusculoskeletal. If we're exercising, it's cardiorespiratory. So to talk about these alveoli and gas exchange, we have to take a look at the cardiorespiratory systems. There's your heart. Your lungs containing millions of alveoli, the functional units in the lungs. The heart sends deoxygenated blood to the lungs. Gas exchange occurs there, and the oxygenated blood returns to the heart. So if we blow this little area up, deoxygenated blood moving toward the alveoli, oxygenated blood moving away from the alveoli. So this is from the heart. This is to the heart. Why are the alveoli filled with oxygen? Well, there's oxygen in the atmosphere. We inhale. We bring the oxygen from the atmosphere into the lungs, into the alveoli. So now, what specifically do we mean when we say that's the lung, the functional units are the alveoli, they're made of simple squamous, the function is gas exchange. Gas exchange. What gas is going where? What gas is moving from the alveoli into the blood? Oxygen. Oxygen. Is there another gas moving?
Yep.
what?
Carbon dioxide.
Carbon dioxide, and the carbon dioxide is going from where to where?
The blood.
From the blood into the alveoli, and then we blow the carbon dioxide off into the atmosphere. It's not difficult, but make sure you know what's meant by gas exchange. Now, of course, what's the purpose of all of this? Well, the heart pumps the blood to the lungs for the purpose of oxygenating the blood, but why? Why does it do all of that? Well, because the heart has to also pump the oxygenated blood to all of the cells and tissues of the body to oxygenate them. So down here, you have to oxygenate your brain, your intestines, your toe. Every part of you must be oxygenated. And we pump the deoxygenated blood back to the heart. Okay, these chambers, you've certainly heard of them in elementary school, but this is your left atrium, left ventricle, right atrium, right ventricle. So just for completeness, I would probably add those labels. So in terms of simple squamous, name two organs where we've seen simple squamous. Kidney and lungs. Name three structures where we've seen simple squamous. Alveoli, Bowman's capsule, glomerulus. You get the difference between the question. Organ, structure. The structures are typically the functional units of the organ. So we've seen simple squamous in three structures, two organs, three structures. But I bet you found a lot of other locations for simple squamous, didn't you, in the histology supplement or the lab manual? So you wrote those down, huh, in your histology books, other locations for, yeah, give me a couple.
Your academics, right?
That's not simple, that's stratified. The endothelium, okay.
Mesothelium.
The what? Mesothelium. Capillaries, okay, good. And you got that information where? The supplement? Or the textbook maybe? I don't know. Supplement, okay, good. And that's the kind of stuff, you want to make sure that you built into those books. The second tissue is simple cuboidal. We've seen that so far in one organ. We've seen it in one structure. What am I talking about? Kidney tubules. Okay. Simple cuboidal occurs in, well, it occurs in lots of places. But simple cuboidal, as far as the pictures we have, shows up in another place. What organ is this? Thyroid gland, okay. And what are these structures within the thyroid gland? Follicles, okay. And so the follicular walls, so here's a follicle, so there's a follicular wall. So the follicular wall is made of simple cuboidal. Can't see the cells here, the magnification's not great enough. But if I increase the magnification, yeah, so I increase it, and I'm going to increase it one more time. Now I can start to pick out individual cells. So there's a layer of cells over here that are part of this follicle. There's another follicle over here. There's a layer of cells that belong to that follicle, and I can see nuclei within each of the cells. So thyroid gland, thyroid follicles. These thyroid follicles make a hormone called thyroxine, or T3 and T4. Thyroxine is a glycoprotein. Now in your histology note cards, books, whatever you've got there, you know that I talk about low versus high. It's simple cuboidal, yeah, but low versus high. When the cuboidal is designated as low, the cells look squamous. And when it's designated as high, the cells look cuboidal. But we call it simple cuboidal the whole time. And what's happening is, when the cells become active, they get thicker. When they're less active, they're thinner. Active, I'm talking about production of this stuff. It's kind of like if you have a factory and you make tents. This is your factory. You can fit 25 sewing machines in here. 25 people sitting at the 25 sewing machines constructing tents. Well, if business starts booming and you have to hire 50 more people and buy 50 more sewing machines so that instead of 25, you've now got 75, you need a bigger room, right? To accommodate those extra sewing machines and the people that are operating them. So as the thyroid gland begins making the thyroid hormone, the cells get thicker to accommodate the extra organelles needed to make the thyroid hormone. What organelles would we need here? Protein. We think ribosome. We think rough endoplasmic reticulum. But it's a glycoprotein. There's another organelle in there. Don't you tell me, because I told you on Monday. Good. Shoot, I know I pointed at it. There's another organelle in there where after a protein is made, the protein can be modified. A sugar group can be attached, or a lipid group. So in this case, it's a sugar group. What organelle am I talking about? The Golgi apparatus. So as these cells that make the thyroxine become more active, they get bigger because they need more ribosomes, more Golgi, more rough. And is this not cool, guys? This is how it works. Do you really want to go out in the parking lot and have somebody say, that's the air filter, that's a spark plug? Aren't you going to tell me what it does? And what happens if it doesn't work? No, I'm not. There's no physiology here, it's just anatomy. That would suck, man. That would suck. Okay, the thyroid gland does make other hormones, but they're a topic for another day. As I told one of you in the lab on Monday, it's pretty difficult to look at cells and in other words, at our very limited magnifications with the light microscope, it's pretty difficult to look at cells and classify them as low versus high. It's more of a I need to know thing than being able to do it. Having said that, if I focus on those cells right there, they don't look very cuboidal. They look pretty thin. They don't look square shaped. If I come over here, there's a couple of cells over here that look pretty squarish, so more like a high cuboidal. But yeah, these, I can't see them when I get up that close. These look pretty thin, more squamous, so probably more like a low cuboidal. But I generally don't ask, is this high or low? It's a conceptual type of thing to understand rather than an identification type of thing. All right, so always as you're studying, whether you're going through your histology documentation or watching the DVD, always recapping, making sure that you've got it straight in your head. So at this point, we've seen squamous in 2 organs, lungs and kidney, 3 structures, alveoli, glomerulus, Bowman's capsule. We've seen cuboidal in 2 organs, thyroid gland and kidney. We've seen cuboidal in 2 structures. They are the follicles and kidney tubules. Okay. Our next tissue is simple columnar. For simple columnar, we go to the small intestine. This is a cross-section of the small intestine. It's convoluted into villi. The villi are these finger-like projections. So that's a villus. There are 4 layers of this organ, and they have sublayers, which we'll get into next semester. We've got, we'll just call them 1, 2, 3, and 4. You might want to use your Sharpie and put those little labels on your picture. 1, 2, 3, and 4, and we'll define those layers when we get to a diagram here in a minute. So the small intestine is convoluted into these villi to increase the surface area. The lining is primarily the simple columnar, but you've read about different types of cells. For example, is there a type of cell in this simple columnar that secretes something? What's the name of that cell and what does it secrete? Goblet cells, and what do they secrete? Okay. So you've no doubt read about in the supplement, whatever, on the DVD. There are different types of cells. It's not pure simple columnar. The small intestine is all about absorbing what you eat. Absorbing what you eat. So we'll talk about that, and we'll blow this up gradually. So now we see a few villi. When we get to this magnification, we only see 2 of those layers, what I called 1 and what I called 2. And again, I'll name those layers a little later for you. We blow it up still further. All of this is layer number 2. That's the lamina propria. And then all of this is layer number 1. And that's the mucosa. Of course, that mucosa, if we had the end of this villus, it comes around like that. Simple columnar epithelial tissue, so individual cells are like that, shaped like columns. Again, it's all about absorbing what we eat. So let's revisit biochemistry. Most of you don't sit down at the table and eat glucose and amino acids. Most of you eat macromolecules. Then you digest the macromolecules and you absorb the micromolecules. So, for example, if you ate pasta you would absorb glucose. If you ate lean chicken, in other words, like a boneless, skinless chicken breast baked, you would absorb amino acids. If you ate bacon, you would absorb glycerol and fatty acids. It's your monomer polymer thing or your micro macro thing. So this word absorb, that kind of takes us back to the kidneys and the nephrons. You know what absorb means. I spilled water, I'm going to absorb it with a towel or a paper towel or something. But what does it mean here? Just like what did it mean in the kidneys? Selective reabsorption, permeability, all of that. Well, here absorption means to transport from the lumen of the small intestine into the blood and then carried to the liver. This got kind of small, so if you can't read it. I'm defining absorption in this context as transport from the lumen of the small intestine into the blood and then carried to the liver. Why carried to the liver? Well, here's a great example. We ate pasta, we digested it into glucose. We carried the glucose to the liver. What are we going to do with it? We're going to use it to make glycogen. We store the glycogen in the liver and the muscle. The digestion is both mechanical. You start out by chewing your food, but it's also chemical. The chemical aspect of digestion involves enzymes. So let's take this cross-section of the small intestine now. And blow it up and see how it really works. So here's a villus, and for a point of reference, that point right there is right there. We know that the villi are lined overall with simple columnar. We know there are at least 3 different types of cells there, though, and we'll get to that. So, making those cells look columnar, giving each one a nucleus. So this layer I'll use the same numbers here. We called that number 1 when we were looking at the photomicrograph. This layer is called the mucosa. the tissue is simple columnar. Now, just as with another tissue a little while ago, I tried to make the point between organ versus structure. The lung is an organ, the alveoli are structures. The kidneys are organs, the nephrons are structures. Well, with this, there's a distinction between the name of the layer and the name of the tissue that forms the layer. There are at least three distinct types of cells in that simple columnar. So we have the simple columnar cells. And their function is to absorb the nutrients. So back to this. What does absorption mean? There are goblet cells. I talk about those so much on the DVD, it nauseates me to think about it, so you can fill in the blank for yourself. Somebody tell us, what do they do? What do they secrete? Mucin or mucus? Mucus. What's mucus?
Additional
Mucin plus, okay. I knew everybody knew that. The third type of cell are the enteric cells, and they produce enzymes. Okay, so the digestive process is not complete when molecules find themselves in the lumen of the small intestine. Oh, you've done a lot of digestion, but there's still a little bit of digestion that has to occur in the lumen. So we've got three types of cells there in the mucosa. The next layer, this is the one I previously called number 2. The name of the layer is the lamina propria. The tissue here is a specialized loose connective tissue. So it's loose connective tissue, just like the loose connective tissue that you've got in your slide sets, but it's a specialized loose connective tissue. The specialization is that it's highly vascular. Now we don't see microscopically, because we don't have enough magnification, we don't see in the lamina propria the structures that I need you to understand. We're going to put them in our picture, even though we don't see them in the photomicrograph. You need a little more perspective. There you see a bunch of villi. So here's our lamina propria. We don't see the structures that I need you to see. They're in there. And if we could magnify things, about 150,000 times, we'd see them. But the three big ones that are in there there's a small, I'd color code this stuff. Colors will even be the same next semester when we're in the digestive system and we're building on this. There's a small artery in there. The artery is providing oxygen and nutrients. Now think about that. Take your very, very general histology. Epithelial tissue is avascular, right? It gets its support from the underlying connective tissue. Connective tissue provides not just physical support for the overlying epithelium, but it provides the metabolic support. So there's no blood supply in this epithelial tissue. Its blood supply is in the underlying connective tissue. That's okay, so your skin is the same way. Epidermis, dermis. There's no blood supply in the epidermis, it's epithelial tissue. It gets its blood supply from the underlying dermis because that's connective tissue. So we've got in the lamina propria little arteries providing oxygen and nutrients, little veins removing the carbon dioxide and waste, but the little veins are also, let's get back to this, what does absorb mean? Transport from the lumen of the small intestine, okay, that's in here, into the blood.
That's where all the nutrients and all the micromolecules.
Yes, yes, yes. So the glucose molecule, that's say in here, gets transported across the mucosa into the lamina propria. So these little veins have the products from the digestion of carbohydrates, proteins, and nucleic acids. Now, there's a reason I couldn't just say the products from the digestion of macromolecules. The reason is because it's not all macromolecules. What did I not include here? Products from the digestion of carbs is here, so the rice we ate, the meat, the beans, the protein, the nucleic acids. Why do we have nucleic acids? Well, everything we eat is cellular. I mean, if you eat steak, it's made of cells. If you eat lettuce, it's made of cells. Cells have nuclei. You've got ribosome. Okay, so what's not included here? Lipids. Lipids. They're not in here. They're somewhere else. We'll get there. So products from the digestion of all of this stuff. Back to the definition of absorption. Into the blood and then
To the liver.
there's a third structure in here. And when we're all said and done in May, you will have this picture in your notes at least 3 times. And every time I draw this picture, I will use green for the lacteal. You don't even have to worry about what a lacteal is today. We'll get there. It'll be after Christmas. I want to remind everybody, histology is the foundation of so many systems in the body. We take this forward, not just to AMP2, but to pathophysiology, to whatever disease process you deal with. If you're going to be a paramedic, you won't take pathophysiology, but you'll still take pathological classes. The lacteal contains the products from the digestion of lipids. Why does the fatty material have to be absorbed into a different type of vessel? Well, we're hydrophilic. Fat is hydrophobic. You all know that fat will not dissolve in water. Go home, take a glass of water, put a few drops of oil in it, stir it forever. The oil will never dissolve in the water. We have hydrophobic things in our bodies. They require special care. Anytime an animal's body has to deal with something hydrophobic, it's difficult. It's hard to do because fat doesn't mix in water and we are hydrophilic creatures. Want to see how hydrophilic we are? Go home, put a hamster in a blender, turn it on high, pour it out through some pantyhose, and you'll find the hamster was 50 to 75% water. Do not do that, okay? I said that to make a point. That would be cruel, horrible, and inhumane, and I would fail you. I love animals, but we are 50% to 75% water. So the cholesterol in our bodies, the estrogen in our bodies, any hydrophobic thing in our body requires special care to deal with. Why is the small intestine convoluted into villi?
Increased surface area.
Increases surface area. Let's blow things up further.
Really quick, for the lamina propria, use a specialized what connective tissue?
Loose, specialized loose connective tissue. And the specialization is that it's highly vascular. All right, let's blow things up more.
We're running out of room.
Huh?
We're running out of room.
Yeah, but we've got a little piece right there. It'll work. It'll work. Don't call that. Draw that. Okay, so I just drew four of the cells, greatly enlarged, and I enlarged them to show something on their free surface. No, they're not cilia. I heard it out there, microvilli. So not only is the intestine, the organ, convoluted into villi to increase the surface area, but the cells themselves, their free surface, also called apical, as you know, that apical surface is convoluted into microvilli to further increase the surface area. Now, this is called the brush border. And it's generally too small to see with the light microscope. It's right at the limit of resolution of the light microscope's capability. We know it's there because of the electron microscope. But what happens is, while we can't really see it, we can detect it because if we use a stain that binds a cell membrane, there's a lot more cell membrane up here to be bound than there is down here. So this part of the cell membrane will stain very dark compared to this. We won't see the microvilli, but we'll know it's there, and we call what we do see, anybody know? Brush border. We call it the brush border. This is just an intermediate magnification, but we can still start to get a feel for it. This is just a little darker right along its free surface. A little darker right along its free surface. This is a super, super high resolution picture. I can't say that I see individual microvilli, but I see something there. Absolutely. It looks almost like the bristles of a paintbrush. And that is the brush border. What's this? Name this cell.
Goblet cell.
What does this cell secrete?
Mucin.
What is the product of the secretory product of the indicated cell plus water?
Mucus.
You see the different questions. Name this layer.
Mucosa.
Mucosa. Name this tissue.
Simple.
Name this organ.
Small intestine.
You see the different questions. None of them are difficult, but listening skills are very, very critical. Name this layer.
Lamina propria.
Name Is that what I told you guys? For the name of the layer?
Yeah.
Is it?
Basement membrane in between there.
Okay, I think glad I haven't erased all of this. Yeah, we need to fix this. The name of the layer is the submucosa. The name of the tissue is the lamina propria. Oh, somebody out there cussing under their breath, stop it. You're not perfect. You try to keep up with all this crap in your head. Oh, I am, Larry. I'm gonna be tested on it. Okay, you're right, I'll give you that. All right, lamina propria. What is the lamina propria? It is specialized loose connective tissue. The specialization is highly vascular. I'll tell you what would have been horrible is if I'd erased all that from the board before I caught my mistake. All right. So that's simple columnar. I'm sure everybody's got that.
So when we're talking about said specialized loose connective tissue, can we refer to the other places that have loose connective tissue, or is that a stretch?
Let me make sure I understand your question. So let's say we were looking at this, and I said, give me a location of this tissue other than the location that you're looking at.
Would you say,
You could say large intestine, you could say stomach,
okay,
but you wouldn't want to say superficial fascia, because superficial fascia is pure loose connective tissue. It's not the specialized loose connective tissue known as lamina propria. You see the difference?
yes.
On the other hand, if you were looking at classic loose connective tissue, I said, give me 3 locations of this, and you can include a location where it might be highly specialized. Then you could say, okay, I've got superficial fascia, I've got deep fascia, I've got lamina propria. Like I was telling some people, and I realize only half of you have been to lab, although you've been studying this for weeks, but like I was telling some of the people in lab on Monday, the questions are highly specific. I don't put a picture on the screen and say, just tell me about this. You have to hope you tell me what I want to hear. No, you get highly, highly specific questions. The questions are oral. Okay, they come to you orally. And so you listen very, very carefully, but it's pretty simple stuff. All right, so wrapping up the small intestine, because we've got layer number 1. What's its name? Mucosa. What type of tissue is it? Simple columnar. We've got layer number 2. Submucosa. Name the tissue. Lamina propria. This is layer number 3. The layer is called the muscularis. And the tissue Smooth muscle. So you could tie this into, I believe it was Monday's lecture, prostaglandins. Yes, that was Monday's lecture. One of the functions I gave prostaglandins after we talked about the whole inflammatory response was that they stimulate contractions in smooth muscle tissue. And the specific examples I gave you for that smooth muscle tissue were the intestines, the stomach, and I might have mentioned the uterus. And then the outermost layer, layer number 4, is called the visceral peritoneum, or the serosa. That's the name of the layer, and the tissue is a combination of epithelial tissue and connective tissue. It's a little more complicated than just one main. Okay, I'm just thinking here about how much time I need to finish this, because the big ones in terms of me drawing pictures on the board pretty much behind us. The skin is pretty simple. Fallopian tube takes a few minutes. Trachea takes a few minutes. The ureter is simple. Pancreas is where histology comes up. Now you've covered on your own in building your books the blood sugar histology with the islets of Langerhans. That's discussed in the supplement in plenty of detail, so I don't need to go over that. I'm going to bring some skeletal system homeostasis in there. The connective tissues are very fast. So I think we'll be fine for the examination being on Wednesday. If something happens and we're not, well, then we'll deal with it.